becomes a bit more resistant to the immune system. And then the immune system adapts to it. And this is the stage of equilibrium. At that point, the immune system is keeping the cancer cell under check. But it doesn't translate. These cancer cells, they do not cause a disease because they are still under control. Only when the cancer cells become fully resistant to the immune system, only then we can see the cancer. And this gives you a notion of what's really going on. When we see the cancer patient for the first time, the immune system has already lost the battle. But it didn't lose because due to a weakness. If the cancer patient will contract influenza or pneumonia or whatever, the immune system will eliminate the pathogen and we will heal. So this means that the immune system is perfect. The problem is that the tumors outsmart the immune system. This is the problem. And immune system and the tumor cells, they employ many strategies to evade the immune system. They use different strategies to avoid it directly, and they also recruit the microenvironment, suppressive microenvironment, to inhibit the immune system. So just immune stimulation, the way we did for 100 years, now you understand why it doesn't work, because this is not the problem. In order to harness successfully the immune system, we need to neutralize the tumor escape mechanism. We can also try to elicit a superior response, to reprogram the immune system, to retrain it in order to recognize and eliminate the cancer cells. So if we go back to this slide, the objective here is to go back from escape. Going to equilibrium is okay. Obviously, going to complete elimination would be wonderful. So now let's try to understand how comes, or what is the basis for the immune recognition of cancer. It is very clear why the immune system responds to pathogens. They have different DNA, different proteins, different antigens. Cancer cells have the same DNA, so how comes that it's recognized by the immune system? And over here, Srivastava, it's cut here, and I'm sorry, but Srivastava suggested 20 years ago already that the random passenger mutations, which are accumulated as part of the development of cancer, may alter the DNA and therefore may alter the antigenicity of the cancer cell. And this causes an antigenically unique cancer. Each cancer is unique with this regard. But it was impossible to test it back then because sequencing technology was very, very premature. But the hypothesis was that mutations leading to neoepitopes would lead to recognition by T cells. 20 years later, the genomic technology has really made a quantum leap forward, and today we can do whole genome sequencing, whole exome sequencing, and we can test these passenger mutations and see what is the mutational load. And we indeed can categorize tumors according to the amount of mutations. And by the way, if we look, the tumors with the highest mutational load is melanoma. Just next to melanoma, we see lung cancer, bladder cancer, stomach cancer. As clinicians, this starts to ring a bell. These are exactly the types of tumors that we see clinical results with the current immuno-oncology agents. So mutational load, high mutational load, it increases the chances that a certain mutation would hit the jackpot. We call it the lottery concept. The more tickets you have, the higher the likelihood that you will hit the jackpot. In this case, the jackpot is immunological relevance or immunological significance. Obviously, this is an oversimplification of what's really going on, but as a general concept, you can understand that. And by using all of these genomic technologies, today we know that Srivastava was right 20 years ago. Indeed, all of these, or almost all of these neoepitopes, they arise in non-oncogenic genes. So these are indeed passenger mutations. So now that we understand what's going on, let's try and understand what is the course of the immune system against cancer. So the first stage is collection of the antigens, collection of the immunological information. The information is brought to the lymph nodes, and in the lymph nodes we need to select only the right T cells, the T cells that will work. And this process is called antigen presentation. We need to identify the right cells, to expand them, to activate them, and only then those effector cells, they leave the lymph node, they search for the tumors, and try to recognize it and destroy it. Each one of these processes, if we will be able to improve it, we will be able to harness or to harness the immune system to elicit a superior response against the cancer. So the soldier that we focus on are the T cells. And the T cells are equipped with a specific, with a unique complex. It's called the T cell receptor complex, the TCR. It's a very complicated molecule, complex of molecules. And on one hand, it can identify the target. On the other hand, it transduces the activating signals. As opposed to antibodies that identify whole proteins, T cells, the TCR, doesn't identify the protein itself, the antigen itself. It identifies small bits of it, which are, these are the peptides. And this peptide must be presented in a molecule which is called MHC, major histocompatibility. Only when, and we call this interaction a trimolecular interaction. The TCR will work only if the right peptide is presented in the right MHC. If something here is missing, nothing will work. And now you can understand what is the meaning of an immunogenic mutation. If this is the peptide, it's called, we call it the double key and lock concept. If the mutation increases the interaction of the peptide with the MHC, we will have better presentation of the epitope. If the mutation increases the interaction with the TCR will operate in a superior manner. So now you can understand what is the meaning of an immunogenic mutation. So let's now drill down into these processes, and starting by the area of the antigen presentation. So as I said, the dendritic cells, or the antigen presenting cells, they collect the antigens, they process it into small peptides, and they bring it into the lymph nodes where they encounter a lot of T cells. And they need to identify the right T cells. How do they do it? Only those T cells with the right TCR, they are started like we switch on a car. And this is the signal 1. But a car, the fact that you switch on a car doesn't mean that the car will start going. You need a gas pedal, and this is the signal 2. This is the co-stimulating antibodies, co-stimulating interactions or proteins. Over here, you can see it operating like that. If there is no co-stimulation, by the way, this process is called anergy. The T cell senses that somebody is trying to steal the car, and it engages the immobilizer. Nobody wants to drive a car that doesn't have any brakes. It's quite dangerous. And after the T cell is identified and starts to operate, then the brakes, the co-inhibitory molecules like CTLA4 and PD-1, they start to come into action, as you can see here. The property or the objective of these molecules is to down-modulate T cell clones with potential, with a potential for autoimmunity. So this is why we have them. And these are the soldiers of the immune system, the T cells. Once they become activated, they start searching for tumor cells, and then they destroy them. All of these molecules, the inhibitory molecules, the activating molecules, we collectively call them immune checkpoints. We have inhibitory, we have activating, and all of these can be modulated. Now, talking about the strategy. So we can focus on increasing the training of the immune system in the lymph node. The idea here is that perhaps we are missing some of the anti-neoplastic clones because they are inhibited by those inhibitory molecules. So if we will be able transiently to take off the leg from the brakes, perhaps we'll be able to unmask new clones with anti-neoplastic capacity. The potential danger is that we may also unmask autoimmune clones. So the idea here, by targeting by improving antigen presentation, is to broaden the repertoire of the T cells. The second strategy is to focus on the tumor vicinity, to focus on these tumor escape mechanisms. For example, tumor cells that express PD-L1 or PD-L2 or whatever, in order to actively suppress the immune system. So if we will be able to eliminate those tumor escape mechanisms, the T cells will be able to operate more freely. Now, talking about CTLA4. CTLA4 operates in the lymph nodes. It starts to be activated once, expressed when the T cells are activated, and it delivers inhibitory signals. And you should know that CTLA-4 is our friend. CTLA-4 is very important because if we look at mice who have a knockout for CTLA-4, they die within few weeks from lethal autoimmunity. So it's good that we have CTLA-4. But in order to treat cancer, we want to just transiently block the activity of this molecule, for example, as you can see here. And the concept is that by blocking these inhibitory molecules inside the lymph node, we will be able to broaden the scope of the immune system. And it was shown that this can actually indeed happen. In this specific work or report, 40 melanoma patients were treated with anti-CTLA-4. In 50% of the patients, new anti-neoplastic clones were identified following therapy. So this is exactly the idea. So reprogramming the immune system has the potential to, it has anti-cancer potential. I'm not going to show clinical data. This is going to be discussed by the subsequent speakers. But indeed, it can trigger long-term anti-cancer effects, lasting sometimes even 10 years. This is remarkable for melanoma. But the price for that would be the unmasking of cells with autoimmune potential, and indeed, CTLA-4 blockade triggers autoimmune adverse effects, and we know that they are autoimmune from the early trials. By doing biopsies, we know that these are, in fact, of the autoimmune spectra. CTLA-4 blockade is not an innocent therapy. Between 25 to 30% of the cases, these patients have grade 3-4 toxicity. So let's now move from CTLA-4 to PD-1. So this is the PD-1 axis, and PD-1 has two ligands. It has PDL1 and PDL2, and once it's engaged, it inhibits the downstream signaling of the T cell receptor. So it inhibits the T cells. And if we look again at the mice models, knockout for PD-1, it doesn't cause lethal autoimmunities like CTLA-4. But it does cause immune dysregulation later on in the mice life. So it is consistent with the fact that PD-1 operates less in the lymph node and more within the periphery, the tumor vicinity in our case or in other places in the body. And it can also provide hints on the potential safety profile when PD-1 is blocked. So if this is the model, for example, of T cell with PD-1 infiltrating a metastasis, encountering a tumor cell, if this is the interaction, by blocking PD-1, we eliminate the negative signals, allowing the positive signals to take effect. There are two main PD-1 blocking antibodies. We have nivolumab and pembrolizumab. And I want to show you, again, by using some techniques from our lab, what it looks like when such a checkpoint, and we didn't block here for PD-1, but rather for another checkpoint, we can actually see what it does. So we engineered melanoma cells to be red, and when we incubate, and we can incubate them with T cells from the same patient and look for what it does. Now, what we are now about to see on the left is without an immune checkpoint blocker. On the right is with immune checkpoint blocker. When the melanoma cells die, they turn their color from red to green. So you can see that there is some immune activity when we just incubate the melanoma cells with the T cells. You can see over here they die, over here they die. But when we add, in this case it's a checkpoint that we discovered, but it operates exactly the same when we block with pembrolizumab, for example. The results are robust. The drug itself doesn't kill the melanoma cells. It it enables the lymphocytes to kill the cancer cells. And we see it also in vivo in our patients. So you are all familiar probably with this paper, and I don't want to discuss the regression of the tumor, but rather let's look at the histology. We can see how this brown staining here is for CTLs. You can see the T cells here, they proliferate, they are activated. This is why these metastases are regressing. The PD-1 blockade It enables the immune system to operate, and now this is the standard of care in melanoma, lung cancer, RCC, and lymphoma. And the toxicity of PD-1 blockade is substantially lower than CTLA-4 blockade, and now you understand why. It ranges from about 7% grade 3-4 up to 16%, so roughly 10% of grade 3-4 toxicity as compared to CTLA-4. Now coming to the last aspect of the talk is PD-L1 blockade. So if PD-1 interacts with PD-L1, why not block the ligand itself? And indeed, we already in 2012 saw clinical hints for this effect in melanoma and lung cancer. I'm not going to go into the clinical data, but since we are now talking about blocking the ligand, it seems that the toxicity profile is even superior to PD-1 blockade, but it may be at the expense of lower efficacy. There are no head-to-head comparisons, so we'll just have to wait as the data accumulates. Now, PD-1 and CTLA-4 are not the only checkpoints. There are many other checkpoints which are today undergoing clinical development, as you can see here. I think that with that part, I would like to conclude, and I know that the subsequent speakers are going to talk about pseudoprogression, so I think that this would be a good point to stop. I'll be very happy to take questions if you have.
Thank you so much.